First of all, I'd like to thank the organizers for the invitation, especially Liz Martinez, who made the contact with me. Today I will share with you some of our recent research here at the Federal University of São Paulo. And my research subject is related to the production and the investigation of peptide-based nucleic acid carriers for gene therapy. And more specifically, I will focus my attention on the use of atomic force microscopy and the other correlated tools to unveil, to investigate the structure of these nanoscopic vectors for gene therapy. First, I give you a summary of the talk, of the story we are going to see in the following. I will start my talk with a brief introduction to Trojan peptides, or cell-penetrating peptides. In the following, I will give you some examples of these cell-penetrating peptides, which have a strong potential for therapeutics, for the transportation of the medicines into cells, and in the after, I will discuss a little bit on the main strategies used to formulate these nanoscopic transporters. And here at this step, I think that this will be interesting for the audience, in the sense that I will present some results from atomic force microscopy assays, which have been conducted mainly on Park instruments. And so my relationship with Park systems started a few years ago when I started to investigate these nanoscopic complexes, joining basic science on molecular biology with atomic force microscopy. And in the end of the talk, I will discuss two main examples from these systems. So Trojan peptides, as the name suggests, Trojan peptides are short peptides, short protein sequences, or amino acid sequences, or short pieces of proteins, which have the ability to translocate cell membranes. In other words, we have a few molecules or some examples of proteins that are able to penetrate cells. Since we have molecules with this capacity of penetrating cells, a logical approach that comes from this behavior is to use these molecules to transport exogenous molecules into cells. Here in the right, I give you a few examples of loads which have been reported in literature to be transported by cell-penetrating peptides. So these cargoes include, for example, plasmid DNA, or nucleic acids that are vectorized into the cell, and in this case, it transforms the cell machinery. In its case, the cell express new proteins, or it can be used to correct a misbehavior of the cell, and then it can be used as a therapeutic, in fact. But other examples of cargoes are also found in the literature. For instance, cell-penetrating peptides have been used to transport small parts of proteins into cells, or even inorganic nanoparticles, such as iron beads, for instance. So liposomes, and we have now a very nice example. We are looking the development of this story with the formulation of vaccines based on liposomes. The Pfizer vaccine, for example, is a very nice example of this, where we use a liposome to transport nucleic acid cargo into cells in order to transform the cell machinery to express a kind of protein that will be used to trigger immunological response. So here I give you a few examples of cell-penetrating peptides. These sequences, as I said before, they are characterized by a short number of amino acids. So here we have a few examples with CPPs ranging from only 6 amino acids here up to 24 here, for instance. So most of the sequences are within a range of sizes between 5 and 30 amino acids, and they have a very diversified origin. For instance, many cell-penetrating peptides are derived from proteins, or for instance, they are derived from virus proteins. A classical example I can give you is the so-called TAT-HIV CPP, which is a sequence derived from the HIV virus, for example. So a major characteristic of the sequences is that they are usually cationic sequences, in the sense that they have positive charges, and these positive charges can be used to form complexes with DNA, for instance. I don't know if everybody here is familiar with biochemistry or more molecular biology, but DNA is a very anionic macromolecule. DNA is a really negatively charged system, and in this case, if we have a molecule with the opposite charge, we can use this molecule to form complexes with DNA. So to formulate complexes, or to formulate transporters for gene therapy, for the transportation of DNA, we have two main strategies. The first strategy I show you here is the so-called covalent strategy. This approach consists in the covalent bonding between the cell-penetrating peptide and the DNA sequence, where I make a chemical bond between these two species. This approach yields higher delivery efficiencies, and it has been a primary choice to produce these vectors based on cell-penetrating peptides. However, a few drawbacks emerge from this approach. For instance, since we have a complicated chemistry to make the covalent bonding between peptide and DNA, this is an expensive approach, and also it's hard to prepare. It requires several steps, and it requires a high level of specialization to prepare this system. So in this context is an alternative strategy. This one, the non-covalent strategy, is an interesting alternative. It's interesting because the complexes in this case, they are obtained just by mixing the negative species, the DNA, for instance, and the positive species, putting them together in the same solution. So the opposite charges make the job. They form the complex by simple electrostatic attraction. So this makes the preparation very easy to perform, and in addition, it has a very low cost. It's really cheaper to produce the complexes instead of a conjugate through refined chemistry, for instance. But the drawback in this case is that these complexes, they show a lower delivery efficiency. So in our group, we have asked, we have decided to investigate the reasons behind these lower delivery efficiencies. And we guess, or we hypothesized that the reasons behind the lower efficiency of these molecular carriers could be related to their structure. And in this case, to investigate the structure, we have used, among other tools, atomic force microscopy. So now I start to give you an example of a recent system that we have investigated in our group. This is a Trojan peptide, a cell-penetrant peptide called penetratin. Penetratin is a 16-mer fragment derived from a protein called antennapedia. Antennapedia is a protein that is part of the embryonary cycle of this fly, the Drosophila melanogaster. So this short sequence is able to penetrate the cell, and it's also highly cationic. Almost half of the sequence is composed by charged groups. In this case, it's quite easy to produce complexes with DNA using this peptide. To investigate the structure of complexes based on this peptide, we have used atomic force microscopy, and more specifically, we have combined atomic force microscopy with IR, or infrared spectroscopy. In this case here, I show you a few examples of the technique. I don't know if most of you are familiar with the technique, but essentially the nano IR spectroscopy is based on traditional microscope. We have a cantilever tip scanning the sample, but simultaneously we have a laser pulse, an infrared laser illuminating the surface. In this case, those sites across the surface where we have a larger absorption of infrared radiation, they will suffer these parts, these regions where we have larger infrared absorption. We have a thermal expansion, which is captured by the AFM cantilever. Okay, so the technique allows to produce infrared maps or absorbance maps. For instance, here we have the same region with nanoscopic aggregates, peptide aggregates, but they have been illuminated at different wavelengths. So here we have a topography image, a traditional topography image, but the other three images correspond to scans obtained under infrared illumination. So in this example here, I'm pointing to you, the surface has been illuminated with a laser at 1600 centimeters -1. Here we have 6090 centimeters -1, and here 6030 centimeters -1. These wavelengths, they correspond to absorbances related to the amide band of the peptide, or these wavelengths, they are specific for different conformations of the peptide, and we can observe that depending on the structure I am looking for, I am investigating, I can observe different distributions of these structures across the aggregates. Okay, another possibility offered by these two is the collection of infrared spectra from individual or for single nanoparticles. So here, for instance, I have several spectra collected at different regions of the fiber, for instance, and I can observe the transformation or the change of secondary structure across the fiber. Or I can compare, for instance, the vibrational profile of different polymorphs. Here I can compare, for instance, the infrared signature of a globular nanoparticle with the signature of the fiber fibrillar nanoparticle. So we have used this tool to investigate the peptide DNA complex. Here I show you a topography image of the complexes, which have regular morphology or some a more elongated shape, but without some particular regular shape. And here I show you the absorbance map of these aggregates. More specifically, the wavelength used to obtain this image or this wavelength, it corresponds to the vibration of beta sheets. Okay, so in this case, in red here, I can observe the region or the regions of the particles which are richer in beta sheet conformations. Here I show you some images from the semi-complexes, but now I can compare a specific wavelength related to the peptide fraction or to the formation of beta sheets, for instance, and I can also map, or I can also investigate the distribution of DNA, because now here now in the second image, we are scanning the surface under illumination at a specific vibration corresponding to the DNA. In short, just to summarize, when we superpose these maps, I can, in this case, to see the distribution of the peptide band in green and the distribution, the distribution of the DNA band in green and the distribution here of the peptide band. Okay, so the combination of these two absorbance maps showed to us that the peptide is concentrated in the core of the aggregates, whereas the DNA is around. This is closer to the periphery of the aggregates. In other words, we have shown here that the DNA, its complex, is at the outer surface of the complex. And this makes a huge difference because if the DNA is at the outer surface, this means that this DNA is more exposed to degradation from DNases or from protein from enzymes in the extracellular medium. In this case, we have shown an evidence for the lower efficiency observed in this kind of complexes. Since the DNA is concentrated at the outer surface, this means that we can degrade the DNA easier when we have this kind of approach. This data have been complemented with other techniques. I will not give more details because our focus is on atomic force microscopy. But in short, we have observed that the structure of these aggregates composed by sub-units with globular shapes held together by fibrillar assemblies, making a nanoscopic association that packs the peptide in the inside. So, we have discovered that penetratin, a classical cell-penetrating peptide, is not very good to formulate complexes through the non-covalent strategy because when we do in this way, we have a concentration of peptide in the core of the complex and the DNA remains at the outer surface. So this architecture is not convenient for gene therapy, for instance. Okay, for the reasons I already discussed with you. In this case, we have changed the strategy and we have selected just the core of the penetratin. In other words, we have concentrated our attention. We have just cut the penetratin sequence, and we kept only a small part of the sequence containing most of the hydrophobic amino acids. In other words, this peptide now it's shorter, it's less cationic, but it's more hydrophobic in the central region of the sequence. We have combined this peptide with DNA through the same strategy, and in this case, we have observed that the transfection efficiency increases. That's a really interesting factor because in principle, we would expect that the entire peptide would be more efficient to penetrate the cell. But in this case, we have shown that with only a part of the peptide, we are able to penetrate the cell, and by using atomic force microscopy, we have unveiled the architecture or the organization of the peptide, of the complexes. In this case, we have the DNA in the center of a fibrillar complex, but surrounded by the peptide. In other words, when I change the molecule, I change the strategy, we can obtain an organization where the load, the nucleic acid load, is protected inside in the core of the complex. Okay. So I am closer to the end. I show now another example of a hydrophobic peptide that we have investigated in our group. This is a peptide derived from the alpha-1 antitrypsin. It's also a protein, and it's an hexameric sequence that does not exhibit charged residues. This is an important factor. Well, we have investigated these through different techniques, and essentially we have shown that since this peptide is highly hydrophobic, it forms aggregates at really low concentrations. Okay, I will not concentrate my attention here because I want to show you these nice results from atomic force microscopy assays. The aggregates formed by this peptide, they are fibrillar at the nanoscopic scale, as shown here. And the interesting part is that it's able to form crystalline assemblies, such those ones I'm showing you here. Here on the right, I'm showing some optical microscopy assays that confirm through microscopy, light polarizing microscopy, that confirm through these birefringence patterns the crystalline structure of the aggregates. And it's interesting that the peptide forms fibrillar aggregates at a nanoscopic scale, and over the course of days, these structures they combine and they grow through the micrometer scale, as shown here. We have been able to make complexes with DNA as well. In this case, electrostatic attraction is not a major force driving complexation. We have hydrogen bonds, and we have verified the mechanism of complexation through the combination of atomic force microscopy and infrared spectroscopy. So by investigating the infrared profiles or the vibrational profiles across the complexes, we have been able to establish that they are formed through hydrogen bond. These complexes have been used to penetrate cells or to transport DNA into cells. Here, the DNA appears in green, and essentially, when cells are incubated with these complexes, we are able to penetrate the cells as shown here. And an interesting factor that I would like to highlight just before to finish my talk is that from the combination of this information, most of them provided by atomic force microscopy combined with other techniques, other approaches, of course, we have been able to discriminate the aggregation pathway of this complex and to extract very valuable information that has assisted us to optimize and to produce more efficient complex for gene therapy, for instance. So in conclusion, we have that penetratin is a strong condensing agent, and this is not surprising because since it has many cationic groups, it makes strong interaction with negatively charged DNA. But in this case, a major finding from our analysis is that the DNA remains at the outer surface. And this is one of the reasons, or a very important reason for sure, to explain the lower transfection efficiencies that we observe when we make the complexation through this strategy. Okay, to finish, and I swear that I am finishing. I'd like to acknowledge my research group. These are my students who have made most of the work I've shown here, and these are our sponsors: FAPESP, CNPq. These are Brazilian funding agencies, and if you are interested in our work, I invite you to have a look at recent publications from our group here. I show you 3 publications that have been featured on cover letters, on the covers of international journals that you can find on the internet. And if you want to contact me, I will be happy to discuss and to answer the questions. Armando, thank you very much, and sorry for the time.